Series of Swift Analytical webinars. As you may know by now, my name is Malcolm and I'm responsible for the business development within Swift Analytical, and I'm going to be chairing the webinar this afternoon. I know, as you'll see from the slide, the webinar is being presented by Dr. Joanna Niska-Blakey, and Joanna graduated with a PhD from the FIR Institute in Milan, where she was investigating DNA replication termination challenges in cancer prevention. After graduating, she did postdoc research in Singapore before joining BLIRT as the scientific support manager. And Joanna really has a broad knowledge and understanding of all the associated techniques, the applications, and the scientific trends which really are essential when providing the connection between the requirements of you, the customers, and the experience of BLIRT's application specialists. And today, Joanna will be talking about the importance of reagent selection for developing diagnostic kits. And we'll be focusing on the key products manufactured by BLIRT that really are indispensable in novel testing kit development, and which are now used in a broad range of biomedical diagnostic applications. For those of you who don't know BLIRT, they are a custom-made enzyme manufacturer with over 25 years experience in life sciences. And their motto is enzymes as you need, which really reflects BLIRT's business strategy. And above all, really demonstrates their individual approach to every client, application, and cooperation model. And one of the things this pandemic has really taught us is that by combining forces with reliable partners, it really helps create rapid and appropriate responses to global health issues. And in consequence, of course, saves more lives. As usual, there'll be the opportunity for questions using the Q&A chat box. So please feel free to ask questions. And we'll try to answer as many questions as we can during this webinar, but if not, we'll answer your questions offline as soon as possible. But before we start the main presentation, I'd like to introduce Dr. Meb Survey, who's going to give you a short overview of Swift Analytical for those of you who are not aware already of Swift. Over to you, Mebs.
Thank you, Malcolm, for the introduction, and welcome everybody. Appreciate you very, very busy. So while people are still joining us, I just wanted to spend a few minutes talking to you about Swift Analytical, what we do, and about our capabilities. So I'm just trying to figure out what's happened with my controls. It's just frozen on me. So let me just go to the next slide. Here we go. So my name's Meb Survey. I'm the managing director for Swift Analytical. So the company was established in February 2011. We celebrated our 10th year this year. We're based in the beautiful and historic city of York. We're a family-owned UK distribution company. And what we do is we bring technologies to scientists and researchers in a number of areas, which include single-cell omics, high-resolution bioprinting, and 3D cell culture. We're also proud to be associated with BLIRT, BLIRT and Swift have had an association now for over 5 years. And in that time, we continue to provide high quality, high performance reagents for assays and diagnostics development. So let me just quickly take you through some of our products. So the first product that we have is by our partner in Sweden called Fluicell. And what we have is a liquid handling technology, which is able to precisely deliver reagents to cells or groups of cells with very high precision. And the key attribute here is that we can do this without mixing different zones. So when you're doing single cell omics, you know that you're directly influencing the cell or groups of cells. So the other product that they provide is a high-resolution single-cell bioprinting system. And here is the device. And on the next slide is the output that we are able to generate. So some of the tissue models that have been created so far are liver models and skin cancer models. These models have been laid down with very high precision, low shear rates, and are being used now by a number of large pharmaceutical companies around the world. The next product I'd like to introduce you to is the iWash slide cleaning system. And one of the key attributes here is that it's a cleaning system for cell counter slides, which allows you to reuse slides between 20 and 50 times. It's compatible with all the major players, and it's providing economic as well as environmental benefit to the tune of at least £20,000 per system per year for our customers around the world. The next company I wanted to introduce you to is a company based out of Germany called Biofluidics. And here we've got a very powerful, versatile liquid handling system, which delivers picoliter, nanoliter to microliter volumes with very high precision. So although we've got a 1536 well slide shown on the right-hand side, this can actually be used for developing custom diagnostic kits. So if you've got multiple components that you want to dispense to create your kit, this device will allow you to do this. And if you want any more information, please do not hesitate to contact me after the webinar. The next product is now within our 3D cell culture portfolio. And here we have the Cell Dynamics system, Cell Dynamics weight system. And it's the world's first flow-based device, which allows you to simultaneously measure the weight, mass density, and size of sphere-like biological materials by gravimetric means. The key attribute here is it's a game-changing technology, because in the past, whenever you viewed the spheroids within under a microscope, all you could see is the actual size of the size of the cell, but also you could see that it was dark, but you didn't know why it was getting dark. So with the weight system, you're able to now measure these additional parameters. So if you are doing drug exposure studies, for example, you'll be able to monitor attributes such as the shrinkage, and that will have a direct impact on the drug efficacy. So coupled with this portfolio is a couple of options for cell culture plates. So if you are generating 3D spheroids in large batches, then we have a 5D cell culture plates provided by our partner Kugelmeyers. And within each well, you can generate reproducible spheroids in clusters of 750. If you want to have your spheroids ready for high throughput screening, then our partners in Facilitate will allow you to do this. And here we have ultra low adhesion slides as well as coating solutions. So if you are developing your own chips or technologies, then we have the coating technology to allow you to do this. So now thank you for your attention, and I just wanted to give you an introduction to our headline speaker, Joanna. The company is ISO 9000 2015 certified, and recently I'd like to congratulate them for achieving ISO 13485 accreditation as well. So thank you very much for your attention. I'd like to now hand you over to Joanna.
Good afternoon, everyone. Okay, I just need to share the screen. If you give me a second. Okay, can you see the presentation well? Okay, and you can hear me well as well, yes? So I'm very happy. So thank you very much for the nice introduction, and most of all for the invitation on behalf of BLIRT. It's a great pleasure to be here and present this webinar. And knowing the diversity range of the attendees, I deeply hope that everyone would find at least a part of this webinar informative and useful. Few words of BLIRT, even if it was mentioned by Malcolm and Maps, because some of you might not have heard of the company before. So BLIRT stands for Biolab Innovative Research Technologies, and we are a Polish biotech company. We manufacture enzymes, and as has been mentioned, our motto is enzymes as you need. which very well not only reflects the flexible business strategy, but also very personalized approach to every application, to every client. Importantly, we are certified with ISO 901 as well with ISO 13485. And historically speaking, we are a technical university spin-off founded over 25 years ago. But since 2011, we became publicly listed on the recognized exchange stock. So even if we started very small, almost 3 decades ago, now we are very proud to have a large-scale production up to 20,000 liters. We are still dynamically growing. And just to give you an idea of the size of the company, at the moment, it consists of 68 employees, of which around 1/4 have a PhD degree. We are located in northern Poland and spread into 3 sublocations. So 2 are in Gdańsk, where at the university, as well as a science and technology park, where our offices, our laboratories, R&D department, production are located. But what is important, we have our 3rd sublocation in the nearby city. And the rationale behind such isolation is that we keep our protein escape production there. And the reason behind is to avoid any potential cross-contamination with other reagents. Otherwise, we'll be in a big trouble. So BLIRT actively participated as a reagent supplier during COVID-19 pandemic. And what we've learned during the last 16 months is that diagnostic tests are very important. And only adequate and rapid diagnostics can lead to the optimized patient care and subsequently prevent future infections. And the second lesson that we've learned, which is strictly related to the first one, is that we only perform better when we join forces. So only collaborations, coordinated response can lead to really technological breakthroughs. We all are aware of the consequences of the lack of diagnosis, which happen both on the individual as well as a social level. So the lack of treatment can lead, obviously, to a disease, which can be easily transmitted. But equally dangerous is the treatment based only on symptoms, because it can lead to mis or over-treatment, and furthermore, to drug resistance and waste of resources. So it is not surprising that we all observed an increased demand for diagnostic testing during the last 16 months. And even if BLIRT is an enzyme manufacturer, we do not generate diagnostic tests. We have learned from our clients that such tests develop, that there are three very important factors to be considered by every startup, by every entrepreneur. The first is you need to think well about the type of biological sample based on disease or condition that you would like to diagnose. Once you choose the sample, you think of the adequate type of test, and consequently, you think of reagents. So most commonly tested samples in diagnostic tests are these ones presented in the picture due to the easiness of their collection. And there are hair, saliva, sputum, blood, semen, urine, or tissue. When it comes to the choice of tests, you need to fulfill certain criteria, which are developed by the WHO Health Organization. And they're listed in this assured list. And it says that tests ideally should be affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, and deliverable to end users. There are different types of tests. You have antigen-based tests, you have molecular-based tests. And the reagents that we produce as an enzyme manufacturer are specifically used for nucleic acid-based diagnostic tests. And on the next slides, I'm going to show you a few types of methods, like applications that our reagents are used for, and certain tests that our clients use to develop their own, let's say, on diagnostic tests. So number one, which is widely used in diagnostics, is RT-qPCR. And as you see, we do produce reagents that are used at every single step, starting from sample preparation, finishing on DNA amplification, such as proteinase K, isolation kits. We try to increase the sensitivity of tests by, for example, producing RNase inhibitor. We do produce different types of nucleases, including DNase-me, which is a type of DNase-1, and obviously steps as reverse transcription and with our reverse transcriptase and DNA amplification using our Technova polymerase. But what is very important, what we heard from our clients when you're a startup and you think of developing a test, is to be focused on certain kinds. And the reason why is to narrow down the list of reagents that you can use. So this is another method used in diagnostics, which is RT-LAMP. It's quicker than RT-qPCR, and it doesn't need sophisticated instrumentation. But when you look at the reagents, they're very similar, if not the same, as an RT-qPCR, obviously, except of the final, like during the DNA amplification, you use BST DNA polymerase. And the third method, I think it's worth showing, just to present you that our reagents are used in relatively novel methods, novel techniques, which is in situ sequencing, with a massive potential in cancer diagnostics, because you're sequencing mRNA directly from the tissue. And this tissue is like, it's in a section, like it's in a section, yes, on a glass. So what do you do? In our workflow, we are producing reagents starting from reverse transcription until the very end as a sequence by ligation. And again, you're using our reverse transcriptase, RNase inhibitor, but also different reagents as TTH ligase, T4DNA ligase, P29 DNA polymerases. And what is important from this method is that it provides you a big amount of data, a big amount of information, and potentially it can even show the drug target. So once you choose the test, the type of test, you need to think of the reagents. And this is a very important and crucial part for all startups, for our companies that think of such businesses. So we differentiated for main criteria, I would say, which is the high quality, stability, reagent compatibility, and availability. And each criteria has its own sub-criteria. And I'm going to go group by group by giving the real life examples based on our experience, on our BLIRT products. So the first category, the first criteria is the high quality. And by high quality, we mean sensitivity and specificity of the reagents. And here I will be talking about importance of being certified, quality comparison to market leaders, batch-to-batch consistency, and activity in stringent conditions. So quality defines sensitivity, and sensitivity is guaranteed by accredited certifications. So what has been mentioned by Malcolm and MEPS, BLIRT was accredited to ISO 13485, which entirely covers the activity of the company, which includes designing services, development, production, sale of certain reagents used in IVD tests, also biotech processes. But what is an important note that I would like to highlight here for all IVD manufacturers is that new regulations come into life starting from May 2022, which means that such manufacturers will need better documentation, traceability of the reagents that they purchase. They will need to minimize certain risks as well. They will need greater product quality control. And this is the reason why it's always worth to think ahead and try to find a supplier which is ISO 13485 accredited. The other very important feature of the products, as you are an enzyme manufacturer, you need your product to be to be as good at certain characteristics as good as the equivalent competitors. So we always compare certain features to our competitors. In this case, you can see that on the graph, the proteinase case activity is compared with 4 different suppliers. And the blue line represents declared minimum activity, which is 30 units per milligram. But what is the important message here for every startup is that when you're looking for a supplier, you should at least look for 2 or 3. You should test certain product at your conditions. And ideally, you should find a product which is interchangeable with competitors. In this way, you assure the continuity of the supply chain. And when you need to replace one product with the other, you're sure that your assay, the sensitivity of your assay will stay exactly the same. The other feature I would like to mention today is the reproducibility, which is very important. And we always check batch to batch possible variabilities. And you see, when looking at the proteinase K, again, activity, there's very low batch to batch variability, meaning that the product is very consistent. It has been done internally, but what's most important for us and is the best sign that we're doing things right is the type of feedback we get from our clients. So this is an email we've got from our client who was testing the activity of our RNase inhibitor. And he tested from 3 separate batches, comparing it to the competitor's one. And you see that the variability is very low, and but the satisfaction of the client was very high at the same time. So this is the main message we always would like to receive. The last subcategory from the quality group is the activity in stringent conditions. But what I want to say here is that we always try to get products with certain distinct, outstanding feature that make our product in a way more attractive than the others that are available on the market. So the very good example is our DNase Me, which is a DNase 1, equivalent of DNase 1, so a double-stranded DNase. And what we've done here, we compare its digestion capability at a broad range of temperatures, and we compared to our competitor, which is the supplier one, with the proper control on the right. So our DNase Me works at a very broad range of temperatures, starting from 0 up to 80 degrees, but on the gel you see the minimum 0, the maximum 60. And I hope you can see it very well on your screens, but when you look carefully, DNA is totally digested at 60 degrees, while the supplier's A1 is not. But what's very important, especially when you work with RNA and you care about protection of RNA, at 0 degrees, we also have a total digestion, unlike the supplier A's, where still the product is partially digested only, which is a very big plus and benefit that makes the product very, very, as I said, attractive. The other group, the other category that I would like to talk today about is stability. And stability during transport at different temperatures, starting from RT at 37, 50 degrees, during multiple freeze-thaw cycles, and also stability of products with different formulation. And I will be talking about lyophilized products as well as lyo-ready. And I will explain in details what these products are. Stability during transport is important because it's cost effective. It's always cheaper to ship a product without dry ice, for instance. And also we worry less about certain accidents at the customs when the package is stuck. So here you can see 2 separate graphs. On the left, there's a proteinase K activity presented, and the relative protein content is compared to its activity at room temperature. And the experiment was done up to 13 months. It's still ongoing, but you see that there is no change in the protein content, no change in with the activity. On the right, there is a graph that represents activity of proteinase K at 4 degrees, which is depicted in orange, and 37 degrees in blue. And we only see it until 18 months. Again, the research is in progress, but we do not see any changes in activity, which is a big plus for the product. Stability at room temperature is one thing, but also improved stability at higher temperatures is very beneficial for two separate reasons. First of all, you can carry processes at higher temperatures. And when you carry processes at higher temperatures, there is no need to react reagents, which again is cost efficient. And as an example, I would like to show our RNase inhibitor and its activity presented at 37 degrees and 50 degrees. So you see that at 37 degrees, it's very stable, even up to 4 weeks. It remains 100% of activity. But when you looked at the RNase inhibitor at 50 degrees, it's stable up to 24 hours. And this experiment was done when RNA was incubated with RNase A, and obviously with the RiboProtect 40 units per microliter. No one likes the freeze-thaw cycles, and everyone tries to avoid it. Sometimes we cannot. Sometimes it's accidental, or as I said, we cannot simply avoid it from the experimental point of view. And it's also important to know whether, in a way, it affects the stability of the protein. So here again, I come back to the example of RNase inhibitor. So RNA was incubated at 37 degrees with the presence of RNase A, an inhibitor, and inhibitor at different stage of cycling, yes, freeze-thaw cycle. And it was tested from cycle 0 to cycle 10 with the proper controls. And there's no difference in activity of RiboProtect. What you can see by the intact RNA, you can see 2 nice bands, which means that the integrity of RNA was protected. The real game changer in molecular diagnostics is lyophilization, as well as we hear more from our clients, the interest is higher in the lyo-ready products. So what exactly is lyophilization? So it's just freezing the product in a solution, placing it under the vacuum, and removing the ice through sublimation. And such products are stable at RT. They're cost efficient because of it. And what's most important, you keep the sensitivity after reconstitution. And there is a nowadays trend of the lyophilized nucleic acid-based molecular point-of-care assays. And such very good examples are RT-LAMP test. And the other very important product, as I said, the demand becomes very high, are the lyo-ready reagents. And these reagents are supplied in a buffer. I mean, they're resuspended, yes, in a buffer which does not contain glycerol. And we all know that glycerol is important because it keeps the protein stable. So from the production point of view, it's very challenging to develop such reagents that are stable during storage. And again, after lyophilization and reconstitution, they still remain their sensitivity. So this is just coming back to the lyophilized products. I would like to show you the graph. This is an RT-qPCR reaction where we compare same product. It's a reverse transcriptase, which we call, it is our transcript product, where we compare the standard version, so liquid one, with the version which was lyophilized and then reconstituted. So what has been done, the serial dilution of RNA was subjected to reverse transcription with these 2 forms of product, the lyophilized version and reconstituted, and the standard one. And what you see is that the efficiency of the reaction is exactly the same. And I can tell you that our clients use the lyo version successfully in their diagnostic tests. Just to give you an example of the lyo-ready products, because here the situation is a little bit different. So our very good lyo-ready product is the RiboProtect RNase inhibitor. So when we want to compare its sensitivity after lyophilization and reconstitution, what did we do? We took 2 batches of the LIO-ready RNase inhibitor. We lyophilized it, reconstituted it, and to make, let's say, the conditions more stringent, we incubated the products at 50 degrees up to 6 hours. And we compared these 2 batches, lyophilized, reconstituted, at 50 degrees for 6 hours in the presence with RNase A. We compared it with our standard version of the RNase inhibitor. And you see, when you look at the RNA, again, its integrity remains, meaning that there is no change in sensitivity after reconstitution, even at higher temperatures, which is a very good sign. The next very important feature of all products is the compatibility of the reagents. And compatibility on 2 different levels. First, with additives, and the other type of compatibility is the cross-compatibility, so reagent compatibility. So just to give you an example of the compatibility with various additives. So when you have the LIO-ready RNase inhibitor, just an example, very often you don't lyophilize the product by itself. You add certain chemicals, other reagents that you lyophilize it with, and then reconstitute. And very often it's mannitol, it's sorbitol, sucrose, twin-20, phenylalanine. So what did we do? We let's say, added different types, different amounts of certain chemicals to our RNase inhibitor LIO-ready version. We lyophilized, reconstituted, and we checked the RNA integrity in the presence of RNase A, obviously. And when you look at the gel, all RNA bands are intact, meaning that RNase inhibitor remains fully active after lyophilization reconstitution with or without various additives. And this is a very important information for all of you that think of developing your own test, which is test optimization. And what I mentioned, the cross-compatibility of reagents. So this graph comes from one of our clients who decided to check the compatibility of our RNase inhibitor in a LIO-ready version, our transcript-me reverse transcriptase, lyophilized, with the addition of BST polymerase, because he created this reaction mixture first to check by RT-qPCR to check the sensitivity after lyophilization and reconstitution, and then he wanted to create his own RT-lamp method. And he compared these reagents before and after lyophilization, and he did not see any difference. And what I can tell you right now, he's successfully using these reagents in the RT-lamp assay that he developed. It's very important to stay focused. So when you're an enzyme manufacturer, when you're a startup, you already manufacture certain types of reagents. It is very important to keep in mind that you do not need to manufacture all reagents that you need for certain reaction, that you need to develop certain tests. And sometimes, mostly because of, let's say, resources, or you don't have such big manufacturing capacity. So it's very important to know that you can get certain reagents from trusted supplier, reagents such DNA and RNA isolation kits, certain PCR mixes, RT-qPCR mixes, whatever really you need. But you need to define, you need to prioritize what you would like to do by yourself and what other reagents you can get from outside. And last but not least, important criteria in reagent selection for diagnostic kits is availability. And availability when it comes to the amount of the product, availability of the supplier, product adjustment, and the quality agreement. So it is very important for a customer to always look for a supplier with a large-scale production, because you can be assured that you can get the product in a big amount from the same batch, and that will make certain things easier. But also, on the other hand, when you look for a supplier, you should look for someone that can meet your requirements, that can help you to optimize the products, to develop the test that you think of. So it's very important always to ask for a variety of firms, because in certain applications you might need liquid reagents or lye ready or in powder. Moreover, very often we should also think and ask for certain product adjustment. Let's say if you would like to change the formulation of certain product, buffer it's resuspended, or the way the product is being fermented. So all of these are very important, even not details, but they can strongly affect the specificity of your assay. When you're an enzyme already manufacturer and you have a product developed, but you don't have big enough manufacturing capacity, you can also look for flexible business models and ask for the CMO type of service. And in this way, it's also very beneficial to look for additional services provided, such as aliquoting or labeling of the sample. So when you get the reagent back, it's simply ready to be used. And to finish up with, I would like to give the key takeaway message regarding the relation between the customer, so you, and your potential supplier. So from the moment you contact the supplier, you should always be as detailed as possible about the application you would like to use the reagent for, because it will help. It will help, let's say, to optimize the methods at the end. So you should always ask for a sample, ideally for a few samples coming from separate batches, as I highlighted at the beginning. In the meantime, it's very important and beneficial to think of signing the no disclosure agreement. And when you have the first results, you should realize what type of adjustments could be done that can meet better your requirements. And it's about, for instance, certificate of analysis. It's about certain adjustments in the product development and evaluation. So all these things should be carefully discussed with the supplier. When you are happy with the final product, you sign the final approval, and again, ideally, supply and quality agreements, just to be sure that you will be assured the stable supply chain, and also during the time of the agreement, no changes in the certificate of analysis will be done. So it's all to keep you on the safe side. And what's not presented on this outline, from now on, you can happily run your business, hopefully successfully. So I think this is what I would like. This is what I wanted to talk to you about today. And you know, people say that sky is the limit, and with the footprints on the moon, we think that the limits are much further. And at BLIRT, we really believe that when you are backed by bold ideas, by determination, and when you have the right collaborators beside, you can truly take off for space. And it doesn't really matter if you are a startup or a blue chip company. So this is all I wish you all. And I will be very happy to take some questions if you have any. Thank you very much.
Well, brilliant. Thank you, Joanna. That lots to think about from that presentation there. But first of all, an apology that we seem to have had a problem with Zoom allowing some attendees come in. We've been picking up messages for people trying to join. So, but Zoom and the security has kept them out. But the good news is this is being recorded, so it will go online later. So it will be available to go over. And for those who weren't able to join us, they'll be able to pick it up. And then, of course, they can send questions through later. But apologies for that. Technology was kind of defeating MEBS at the time there. We've had a couple of questions come in. One from Susan saying, can you supply already aliquoted reagents?
Yes, definitely. We can supply aliquoted reagents. This is one of our additional services that we offer. So we aliquot, we portion, we label. This is what we do. Yes.
Yeah, I mean, I think the main point is just tell us what you want. And
Yeah, so very often, let's say certain clients can send us even the vials, the tubes, so we can aliquot in the vials that they prefer to have the reagent in. So it's not an issue, even the batch booking. So these are the all details I talked about, that as a supplier, we are very customer friendly in a way. We try to do our best to meet the requirements.
a question from Lars, obviously the new IVDR regulations are probably going to be scaring quite a lot of people. And that's going to affect the development of diagnostic kits, I'm sure. But he's saying, could you just go over that part again?
Yes, yes. So there are new regulations that come into life, I said, May 2022. And these new regulations are about different classification. So there are going to be classes with more stringent classes due to the risk, yes, assessment. So this is one thing. The other thing is that there are going to be more stringent surveillance by the notified bodies. So in general, the regulations will be beneficial because more transparent regulations for patients' safety are going to come into life, which means more control, greater responsibility for the manufacturer of IVD. But as a company, we do not produce CE mark products because we are not IVD manufacturer. But from the IVD manufacturer point of view, it's always safer to get the products for a supplier as BLIRT, for instance, because we can provide full documentation and traceability, and we can minimize the risk as much as possible, providing the greater quality control. So I guess the topic is very broad, and if you have more questions on, or if you would like to know the details, I can always refer you to our quality control director, and I'm pretty sure he'll be very happy to answer all your questions.
But basically, it is going to be more difficult to develop diagnostic kits.
I imagine more challenging, yes.
Another question that's just come in. Is there a job placement vacancy for a biotechnology BSc graduate?
Oh, I guess this is another question to me.
We can talk about that later, but
yes.
no, I mean, certainly any vacancies will be publicised.
I'm happy people are interested in to work at BLIRT as well.
Yeah, no, I'm sure it's a good company to work for. A couple of questions come in directly through to me. But one of the big questions is, how do you see diagnostics developing?
I will give you maybe two answers. Obviously, I mean, during the last 16 months, during the pandemic, we see how important the rapid and adequate tests are, especially now when more and more people have been vaccinated. We see the importance of molecular diagnostics compared to, for instance, antigen-based tests, which can be very ambiguous. So definitely the way to detect infectious diseases, to stop the transmission of the disease is very important. This is something that people will be working on. But, you know, I was working for a decade in cancer research. So for me, what is happening now in cancer diagnostic is equally important. And if you ask me what I personally think in terms of future of diagnostics is our liquid biopsies. So the biopsies, when you can detect the circulating tumor DNA by methods as ddPCR or NGS sequencing. And I think this is the future when it comes to cancer research. But again, when we think of infectious diseases, the rapidness and accuracy is the key.
Yeah. When you look at how things have developed so quickly over a short space of time, and especially COVID's a great example.
Yes.
That's part of the introduction. It's just that partnership, everybody working together to make things happen quicker.
I mean, all happened because of collaborations. So this is beautiful. And how quickly we could have accomplished what we accomplished.
And one of the other things really I picked up there is that you can actually optimize the kits with a customer. I help them optimize their assays.
So we are not developing kits. I highlighted it at the beginning because we do not produce CE mark products. However, we can help to adjust the enzymes that the client might need for his own use, yes, to develop his own test. So we can offer different types of services, consulting as well, like a scientific consulting. So we can help from a different, let's say different side, but we do not develop kits, just the enzymes to manufacture, because I don't want people to be confused, yes? Because as I highlighted, we don't have the CE mark products.
No, I was probably phrased it wrong. He's actually helping them optimize their kits. Yeah.
Yes, I mean, definitely. I mean, we can always, what I was also mention about cross compatibility of the reagents. Yes. So we can work on certain formulation to make it work better, to make it more compatible. Definitely, yes.
Yeah.
In this way, yes.
It's covering everything from the development stage all the way through to the bulk manufacturing.
Yes.
Because that's got its own challenges, but the capabilities are there. Brilliant. I can't see any more questions coming in from anyone. So if you do have any questions following this, then please contact Swift Analytical and we'll do our best to answer the questions. If you want to know more about the process, again, please get in touch. I say this will be going up online as soon as it's been edited, so you can review it again. And in the meantime, I'd just like to say a big thanks again to Joanna for the excellent presentation there, and it's lots to think about. And I hope it's made you think about your own processes and how you develop your own products. But more information can be found on the BLIRT or the Swift websites. So thank you for the time, and we look forward to seeing you at the next webinar. Thank you, and
thank you very much, everyone. Thank you very much.
Bye-bye.
Have a good afternoon.
Bye.